Every second, millions of microscopic reactions take place inside a dairy cow. She takes in red blood, yet outputs a chemically distinct white fluid. The udder serves as the site for this conversion, taking the raw ingredients of one substance to synthesize an entirely different one. Inside the mammary gland, specialized cells manufacture milk drop by drop, building the fluid structure through a series of precise metabolic steps. The scale of this operation requires 400 to 500 liters of blood to circulate through the udder for every single liter of milk produced. This high-volume circulation functions as a biological conveyor belt, delivering glucose, amino acids, fatty acids, vitamins, and minerals. Milk is the result of this massive circulatory and metabolic effort, engineered from the bottom up. The actual production takes place within millions of microscopic, bulb-like structures called alveoli. To supply these structures, a network of tiny blood vessels called capillaries wraps around each alveolus, delivering raw materials directly to the cellular wall. These specialized mammary epithelial cells form the inner lining of the alveolus and drive the synthesis process. These cells pull glucose from the passing bloodstream to begin manufacturing. Inside the cell, enzymes restructure the glucose into lactose, a sugar unique to milk. Lactose acts as an osmotic magnet, drawing water into the mixture and determining the cow's total milk yield. At the same time, amino acids arrive via the capillaries to fuel the protein assembly line. The epithelial cells sequence these acids into milk proteins like casein and whey, building the specific nutritional profile required by a calf. By stripping down blood components and rebuilding them, the cells create a nutrient-dense fluid with a chemical signature unlike the blood it came from. While sugars and proteins are synthesized through one path, milk fat follows a different biological route. The mammary gland harvests fatty acids from the blood, but also incorporates acetate and butyrate produced during the cow's digestive fermentation. Inside the mammary cells, these elements transform into microscopic lipid droplets that become suspended evenly throughout the surrounding fluid. Precise amounts of calcium, phosphorus, and potassium are transported into the mix under strict regulation. Vitamins and immune proteins are added next, completing the recipe for the calf's development. Finally, a massive influx of water dilutes these concentrated ingredients, forming about 87% of the final product. The completed milk is released from the cells into the hollow center of the alveolus, where it is stored. These parallel manufacturing lines unify fragmented blood components into a life-sustaining biological fluid. The molecular assembly concludes when the stored milk is ready for harvest. When the cow is milked, the hormone oxytocin is released, signaling the physical transition of the fluid. Tiny muscle cells surrounding the alveoli contract in response to the hormone, physically squeezing the structures. This pressure forces the milk out of the alveoli, through a network of ducts, into the cisterns, and finally out of the udder. This is the process behind every glass of milk poured. The liquid is built molecule by molecule, using the components of the bloodstream to engineer a new substance. Behind the most common agricultural products lies the continuous work of millions of cellular assembly lines.